Right, welcome to part one of transcription. So at the molecular level, a gene is a segment of DNA used to make a functional product. And this could be an RNA that is used directly, or it could be an RNA that leads to the formation of a polypeptide. Transcription is just simply the first step in gene expression, and that is making the ultimate end product. So by the end of this lesson, I hope that you'll be able to describe the organization of a protein encoding gene and its messenger RNA. I want you to be able to outline the three stages of transcription and describe the various functions of RNA transcripts, because there are more than just messenger RNAs. Transcription literally means the act of making a copy. This is kind of an old word, goes all the way back to monks making copies of Bibles. So just simply the act of making a copy. And in that regard, transcription is just that. It's making an RNA sequence from a DNA sequence. Now, the structure of DNA is not altered as a result of this process. So it still continues to store the information, and it isn't damaged or altered in any way. The DNA just simply comes apart, we make the RNA copy, and then it goes right back together. So, as we said, transcription is the first step in an overall process. So when we have structural genes, or genes that encode for a particular protein or polypeptide sequence, transcription is the first step, and that transcription produces what we call a messenger RNA, or for short, mRNA. And the mRNA nucleotide sequence will ultimately determine the amino acid sequence of the polypeptide during a second process that we call translation. Ultimately, that protein, the synthesis of those functional proteins, determines various traits. And sometimes this is a direct trait that you can see. Sometimes it is just part of a system that ultimately will produce a trait. But this is what we call the central dogma of genetics, or some people go so far as to refer to it as the central dogma of biology. The idea that we have DNA, we have transcription and get messenger RNA, and then ultimately we translate that into a polypeptide sequence. So here's a schematic of that. So we start off, we have the DNA, and remember the DNA is where we are storing that information. We can replicate that DNA so that a cell can give its DNA or copies of its DNA to its offspring. But when we get into the process of needing to produce a protein, there is a small segment of that chromosome that we'll refer to as a gene. From that gene, we transcribe a messenger RNA. And a messenger RNA is just simply a copy of that segment of DNA. It is intended to be temporary and is intended to serve the purpose of ultimately producing this polypeptide. And this process of producing the polypeptide is this thing that we call translation. So here, transcription, we're making a copy, and then ultimately we are taking that message and translating it into the protein that it's meant to encode. All right, some of the key concepts here is that the DNA sequence itself is defining what the beginning of a gene is and what the end of a gene is. And it is also this DNA sequence that is regulating the level of RNA synthesis. And the amount of RNA synthesis will ultimately determine the amount of protein that is being made. Another important concept here is that proteins must recognize and act on the DNA in order for transcription to occur. So it's a partnership. The sequences determine where the stop and start is and how much, but it does that via proteins binding those particular sequences. Its overall process is called gene expression, and this will ultimately lead to the final product, which is usually a protein of some sort, and that protein can interact with the environment or the outside of the cell to determine a particular trait. All right, so here we have a schematic of a gene section of DNA. And the first thing here is we're going to go through a whole series of different terminology that we use to describe this gene. So starting off at the beginning here, we have what's called a regulatory sequence, and that is oftentimes sitting next to a promoter. Both of these are working in concert to signal for RNA polymerase to sit down and open up this particular section of DNA and start the transcription process. At the end of the gene, there is something called a terminator. This is the DNA signal which is telling the RNA polymerase to stop making that messenger RNA. So we start up here, we have these regulatory sequences and the promoter that will start producing the messenger RNA, which we can see down here, and it will end at the terminator. Now, looking at the actual message itself, or the messenger RNA, we'll be starting at a 5' sequence at this end. So what we are copying up here is the complementary or antisense strand. And one of the very first things at the 5' end is something called a ribosome binding site. And this is where this messenger RNA will come into contact with a ribosome. And if you recall, a ribosome is responsible for translating this protein or this messenger RNA into a protein. Just downstream of this ribosomal binding site, and I'll oftentimes refer to upstream and downstream, upstream meaning towards the 5' end, we will have a start codon. That start codon is methionine in eukaryotes, or a slight variant of methionine called formal methionine in bacteria. It is always typically the same codon. There's very few exceptions to that. Following that, we'll have a sequence of many codons, however many long that protein needs. And those are 3 nucleotide sequences that specify a particular amino acid. At the end, there will be a stop codon, and the stop codon is literally a sequence that does not have a transfer RNA that will encode for a particular amino acid. Now, in eukaryotes, this messenger RNA will only encode for a particular single polypeptide. Several polypeptides can be made from that one message, but they are the same thing being translated over and over again. There is an exception to this in bacteria, where bacteria can actually have several functional polypeptides made from a single messenger RNA. So in that case, you would have a start codon, many codons, a stop codon, followed by another start codon, many codons, and another stop codon, and on and on. In bacteria, this allows several closely related subunits or closely interacting proteins to all be made at the same time. All right, so here we have a closer look at the starting DNA strand. So here we have a 5' end, and then a 3' end, and here we have the complementary sequence starting here at 3' and going to 5'. The strand that has the sequences that has the codons that are going to be involved in this particular polypeptide, that is something we refer to as the coding strand. So the coding strand will give you a sequence of codons that would lead to a polypeptide if there was a way of getting it from the DNA directly, but we cannot. We have to get it from a messenger RNA. So what we need is a copy of this coding strand. So in order to make a copy of the coding strand, we use the complementary strand, otherwise known as the template strand, because it is acting as the template for this RNA strand. Other words that we can use to describe this is this coding strand might be called the sense strand, meaning that if you were to read it in terms of codons, it would make sense. If you were to try and read the template strand in terms of codons, it would not make sense, so we would call it the antisense. Now, there are transcription factors, which are proteins that will bind to the DNA, which initiate this overall process, and consequently there will be sequences on the messenger RNA itself, which will direct the translation of that. So let's just stay here with transcription. Transcription occurs in 3 different stages. We have the initiation, the elongation, and the termination. The reason why we have it broken up into these 3 steps is they involve a different set of proteins to regulate each step. So here we have the gene in the DNA. As I said at the beginning, there's something called a promoter. And then we have something at the end called a terminator. During the initiation process, transcription factors, which aren't shown in this diagram, will bind to this promoter site, and it will recruit RNA polymerase. And the activity of the polymerase and the transcription factors will open up a bubble in the DNA, allowing for the beginning of a messenger RNA or any type of RNA. So that is one process. Once that process gets initiated, it is solely up to the RNA polymerase. A lot of those transcription factors will fall off, and we move into the elongation or synthesis stage of RNA transcription. So we just motors along the DNA, making a longer and longer transcript. When we reach the termination point, something will cause the RNA polymerase to dissociate from this DNA, and this occurs through a couple of different mechanisms. But reaching this point, it disassembles, and now we have a free RNA, which can go on to do its job. We have the polymerase, which can be used again, and the DNA has been unaltered. Now, over 90% of all genes in our genome encode for a messenger RNA, which will ultimately result in a polypeptide, or 90% is producing a messenger RNA. But there are, in fact, a whole slew of different RNAs. So some of these we've seen before, such as transfer RNAs and ribosomal RNAs. And we'll talk more about these later on.